You're right, it wasn't fine.
So, when we finished up yesterday, or the day before yesterday, we were talking about these two different models of movement through the Golgi apparatus. One of them being the stationary cisternae model, essentially saying that all the cisternae are just sitting where they're at, and things are going to be passed to the next cisternae, or cisterna, via a small vesicle. The alternate to this is that we have the cisternal maturation model that essentially says that we're not passing things through cisterna. Essentially what happens is as we bring things together and we get a cis compartment, eventually that cis compartment will become one of the medial compartments. It will get larger, and as we continue to modify the proteins in there, we will be getting some sort of transition into the medial compartment, and the medial compartment will become the trans compartment, and then eventually the trans compartment will become the trans-Golgi network, and then butt off vesicles to everywhere that needs to go. So we have kind of these two different models of movement through the Golgi, and each one of them, I should also mention just a couple different things. Neither one of these is actually a very complete or workable model, and to tell you the truth, there's at least 4 more models of movement through the Golgi at this moment, because we are still not quite exactly sure how proteins move through the Golgi. Now this, the stationary cisternae model was the model that we thought for the majority of knowing about Golgi. This is how we thought things moved through there. And is about 10 years before I started college that this started to become, well, hey, this doesn't work very well. So what happened is I called it so there's 2 different names for this. The stationary cisternae model or the vesicle shuttle model. Essentially saying the same thing. They're essentially the same model. I had changed it. Apparently I forgot to change this one over. But just know the vesicle shuttle model is the same model as the stationary cisternae model. Essentially using vesicles to shuttle proteins between the cisternae. Now, if you remember from what we're talking about before, so we use COP2 proteins to send things, or COP2 coding proteins, to make the vesicles that are transporting things from the ER to the cis Golgi network, or to the Golgi. And then when we send things back, we're using COP1. All of the vesicles that are moving between the cisternae are COP1, are made using COP1. And so there are no COP2 made vesicles that are traveling between the cisternae. And so the idea was, it just transitions to being COP1 created vesicles between these cisternae. We're not using COP2 anymore. Those are going from the ER to the Golgi, and then we also use it to send them back to the ER, but then we also use them between the cisternae compartments. One other thing I should mention here is that we've known that also we will send things back. It's common for vesicles and proteins to be sent back to the ER. So going from like a medial compartment to a cis compartment. They're going from a trans compartment back to a medial compartment, and then maybe eventually back to a cis compartment. It looks like the reason for this is that, so we need proteins in these other compartments that need to do like the modification of other proteins. Like if we're going to be doing glycosylation, there are enzymes that need to be doing that glycosylation. But those also need to be modified somehow. So oftentimes those will need to get modified in the later compartments, but then they need to end up in the previous compartments to do their work, so they get sent backwards. Does that make sense, or have I confused you completely? Okay, so the point here is that we've known for a long time there are some proteins going backwards. Here's the problem, however, is that as we look at going forward, and the forward transport of these proteins, there's not enough COP1 to actually account for all of the protein movement between compartments. There's more proteins being moved than the COP1 vesicles can account for. Yes? Yes, I'm recording. I did that. Are you feeling like you're going to need to go back and watch this again?
Yes.
Okay. So we're going to need to send. There's not enough that's going. There's not enough vesicles there to be transporting everything. So the idea is then what we came up is this cisternal maturation model. We're actually not transporting all the proteins between the different cisternae. They're actually, the entire cisternae is becoming the next cisternae. We're only using vesicles to send things backwards. When things need to be sent back, then we send them back via vesicles. But the things that are going forward as the entire cisterna matures and becomes the next cisternae. And so this is the cisternal maturation model. However, there's things that this doesn't completely explain as well. For instance, we have tubular networks that actually, small and minor, but there's actually tubular networks that occur between the cisternae. And we believe these to be kind of the result of fusing vesicles. They seem to be somewhat transient, but this isn't really well explained by the cisternal maturation model either. Now, since the point that the cisternal maturation model there's been a modified cisternal maturation model and a few other models that have come up. I'm not going to spend a lot of time with that. The point here really being that we're not exactly sure how stuff gets passed. Just to give you a little illustration of what each of these are, this is the cisternal or the stationary or the vesicular transport model, where as we go forward we have vesicles that go forward and then they're passed to the next cisternae by an additional vesicle that passes it to the next cisterna or the next compartment. And then occasionally also we have things being sent back as well. In the cisternal maturation model, that's what we have up here. We have vesicles that come off the ER and they essentially fuse into the first cis compartments, but then that cis compartment becomes the next compartment, which becomes the next compartment, and then all we're doing is sending things back. The vesicles that we are seeing between the cisternal compartments are sending things back to previous compartments. Are there any questions on that? Yes?
If they're just moving compartments, what would tell them to stop being a compartment and go into individual vesicles?
To go backwards?
No,
or when we get here?
Stop being the Golgi apparatus.
Oh, at this point? I was talking about that a little bit more. Yeah. Essentially, it comes down to you get the finished proteins. The finished proteins are marked for where they need to go, and you start getting them aggregating and budding at the membrane, and you start getting transport of them away, which indirectly. So it's not like one signal that says the entire thing needs to break up. It's not like, hey, trans-Golgi network, you need to start breaking up now at this point. It's more that each of these individual groups of proteins are like, okay, now at this point we need to send it to the membrane. We need to send it to a lysosome or something else like this. And so that buds off. As each of those individual groups of proteins begin going where they need to go, it leads to the breakup of the trans-Golgi network. Does that make sense?
Is that like a time thing or like a proximity thing?
It's not a time or proximity thing. It is that the signal is complete to send a protein to a certain place. Let me get a little bit.
The process, it just wasn't.
Yeah. So imagine you have a signal that needs 5 parts to be complete, right? On a protein. I'll come back to more specifics here. You had some of the parts here. You said some of the parts here, but then here in the trans-Golgi network, you begin adding the final components that says, okay, now this needs to go here. It's like if we're adding the address, we're adding like one part of the address each place, and you can't send it to where it needs to go until you have the complete address. And then when you add like the final, like the street number, like, okay, hey, I can ship this now, so you put it in a box and you send it off. And so that's what happens here is we get the last part of the address essentially. Now it triggers a series of events that leads it to be packaged up and sent off to where it needs to go. And so, and it's all the individual actions of each of these groups of proteins doing that leads to the break of the whole trans-Golgi network. It's not like one signal that says, okay, trans-Golgi network, you need to break up. Does that make sense? Okay. And this, hopefully that makes a little bit more, because I'm going to talk a little bit more about what happens actually right now from what happens at the trans-Golgi network to find and send things off, and hopefully that makes it a little bit more clear what's happening. So let's do that. Let's talk a little bit about what's happening in the trans-Golgi network. This is the final compartment of the Golgi, and this is where essentially we're starting to get the breakup of the final cisternal compartments and sending off all the individual components other places. We have some additional post-translation modification like glycosylation happening here. And we also have transport vesicles leaving the trans-Golgi network going out to the places that they need to go to deliver the proteins where they need to be. At this point, the coding protein is different than the coding proteins that we had before. It's clathrin. Clathrin has a very particular structure. It's actually the coding protein that I showed you earlier as an example of a coding protein. What I showed you earlier in the figure was not COP1 and COP2, it was clathrin. Clathrin forms many individual proteins of clathrin come together and form like a cage-like structure that kind of makes it look like a buckyball, or like maybe not really a soccer ball, but kind of a soccer ball, but if they were all pentagons or hexagons, hexagons. And each individual clathrin protein is what's known as a triskelion. Now I'll show you why it has this essentially 3-arm structure to it. It has 3 heavy chains and 2 light chains. So it's a hexamer. It has 6 different peptides, 3 different heavy chains which are long and form the arms, and then 3 light chains for each that are essentially in the center that hold together each of the 3 heavy chains. And in total, you get 36 of these triskelions forming the entire cage-like structure that is the entire coding protein complement for the vesicles leaving the trans-Golgi network. Hot tea in the snow. Makes me happy.
So the heavier on the outside, easier, so lighter in the...
I'll show you a picture in just a moment. Hopefully that makes more sense. Okay, so let's do that. Okay, so this is the 3 heavy chains here, and we have the 3 light chains in here. Essentially the light chains are serving to hold together the 3 heavy chains, and they form that sort of structure. So does that give you a good, okay. And then each of these comes together with a series of 35 other ones as well, and forms the protein coat on one of these vesicles. To give you another image of how this is, again, this is clathrin here. And again, it's happening essentially the same way that we talked about vesicle formation happening before. There's a couple modifications to how trans-Golgi network transport vesicles are formed, but it's not particularly important for this class. And the main difference here that we're using clathrin instead of COP1 or COP2, it forms the entire coat, but like we have with other coding proteins, as soon as it buds off that vesicle, that begins disassembling. And then those vesicles are transported to where they need to go and then do what they need to do, which we'll talk more about that, specifically how those are transported later as we're talking about cytoskeleton sometime in the middle of next quarter. Okay, so let's talk a moment about how this trafficking happens. How do we actually go about finding out where these are happening? Like where do these go and how do we know where they're supposed to go? So I'm going to give you one example of trafficking of these proteins. And that's going to be ones that are tagged specifically to go to the lysosome. I'm picking the lysosome because we're going to talk about lysosomes next, so it makes kind of sense. And so for ones that are destined for the lysosome, we're going to have, all of those have a glycosyl group put onto them that has a terminal mannose. It could have many other things on it. It could be a chain of like 10 individual monosaccharide residues, but the terminal saccharide on there, or the terminal sugar, is going to be a mannose, and then that mannose gets a phosphate added to it, so it becomes a mannose 6-phosphate. This terminal mannose 6-phosphate is what tags a protein to be destined for the lysosome. So what we have is in the trans-Golgi network, especially on the internal membrane side of the trans-Golgi network, we have receptors that are receptor specific for the mannose 6-phosphate, and then the entire protein by that mannose 6-phosphate is going to bind to those receptors. That's how we go about aggregating these proteins into certain spaces. We have an aggregation of receptors in an area, and as these mannose 6-phosphate residues are binding to those receptors, we are gathering up all the proteins that are destined for the lysosome. Once we get a certain number or a certain frequency of these receptors that are bound, we trigger the formation of a vesicle. And so the vesicle begins to form. We have budding off of that vesicle. Again, this is going to be a clathrin-coated vesicle. And then that is transported to the lysosome, where it binds with the lysosome and dumps its contents into there. Things that are bound for other locations have similar sorts of mechanisms. Really, honestly, we could probably sit down and teach an entire class about memorizing the particular mechanisms about how things end up different places. But there would not be 54 of you in here in that case. Yes?
Does it bind to the lysosome using the mannose 6P also?
Say again?
Does it bind to the lysosome using mannose 6P?
No, so it's not going to actually bind to the lysosome that way. That's what gets it into the vesicle. So what's going to bind it to the lysosome then is a particular set of snares, like a T snare and B snare, that are going to be specific between this vesicle and then what's expressed on the surface of the lysosome. And so it will get caught by the tether proteins, and if it matches those snares, it will then, the membranes will fuse and dump its contents into the lysosome. Any other questions with that? So these things can be destined, when we create proteins in this way, they can be destined for different organelles, and that's grand. But they could also be destined to just be released from the cell in some way. And there's two different major ways in which that can happen. This could happen by a regulatory or regulated secretory pathway, in which essentially some of these molecules, it's going to be something like hormones, enzymes, neurotransmitters go by this, but these are put into vesicles that then leave the trans-Golgi network and then just sit there waiting for some signal to be released from the cell. Oftentimes they will be transported to just below the membrane surface of a certain part of the cell that is required, where it needs to be secreted from. So for instance, what you might see is a cell that comes down like this, and this is like a double violet membrane, and then you'll see a bunch of vesicles that are sitting right beneath the membrane here, essentially just waiting to be dumped out of this membrane. This is essentially a neuron synapse right there. If you want to know more about that, we will pick up at this point in Animal Phys. However, there is also the unregulated secretory pathway as well, where essentially things are just dumped out. We're not holding them in a vesicle getting ready to dump them out into the intercellular space just based on some signal, but we're just dumping out as soon as we make it. And this is the default pathway for any protein that's not otherwise marked for a specific destination. If it has lost its or does not have some sort of destination address on it, it's just going to be dumped out of the cell. So using the post office example, it's like if they get something without an address on it, instead of like returning it to sender, they just dump it out the back of the post office. Actually, the post office being the Golgi, and I guess maybe the town being the cell, they would drive it to the edge of the town and then dump it in a pit. There we go. What's that? Because it's not marked. Because, you know, that's where unmarked things go. That's where all your letters to Santa ended up. Yeah.
So is there anything useful with that, or is it just like junk that it makes?
No, it's not junk. So this is useful. There are a lot of different proteins that need to just be it's called constitutive release, where it just needs to be constantly delivering something. So I'll give you a couple examples of things that do this. For instance, osteoblasts in your bone that are creating bone have to continually make several different enzymes that continue to remodel the actual mineral portion of your bone. Otherwise, it gets brittle and starts falling apart. And so they are constantly making several suites of enzymes that they just constantly dump out into. Also, other things like extracellular matrix proteins like collagen and a few others, depending on where they're at, need to be constantly just re-added to the extracellular space, because they break down at a certain rate. And so you're constantly dumping some of those out at just a certain rate. And so that's being controlled not by some signal that's going to say dump it out at some point. You're controlling that further back by the regulation of how quickly it's synthesized, like how quickly are we making mRNAs to then go on and make this. That's being handled in the nucleus. Or how often are we breaking down mRNA so we can't remake it, or other things like this. So that's happening, the control of that, and how many we make is happening way further back up the line as we're actually controlling how much protein do we make. But then once you make the protein, it's just on this default path just to be dumped out of the cell. Does that kind of answer your question? Okay. Now in both of these cases, I really hate it when like you're down to the end and you're drinking and like you underestimate how much is in there. You throw it back too far and it goes up your nose. Oh. Yeah, not quite the silence. It's just I have a sticky mustache on. Not that way. Okay, so we actually need to get these proteins out, and so these vesicles are bound to release their proteins or their contents into the extracellular space with these things called exosomes. And these could be doing quite a bit of different delivery. We could be transporting membrane components or proteins or other things into the extracellular space that's going to be transported between cells. And so that's good. This could be for intracellular communication, not just actually delivering materials, but actually just trying to transport a signal. So this would be things like hormones or other things like that. Also neurotransmitters are going to be in these vesicles bound to eventually release into the extracellular space of these exosomes. But then also, if we do have some unwanted molecules that we do want to get out, we will also release those that way. So there will be some just releasing of waste this way. those are also going out by exosomes. Let's see if I can get these better. Okay. How embarrassing would that be if I drowned into my own tea in the middle of cell biology? You'd have a story to tell your children then. I once knew this professor who was so dumb. I won't spell it. Okay, any questions before we move on to the next set of similarly related organelles? Anything on the ER or Golgi at all? Yes, Samantha.
Just to clarify, COP1 comes from the Golgi, COP2 comes from the trans, whatever it's called.
It's coming from the ER.
Okay, the area.
Yeah. Yes.
And exosomes are not in the regulatory secreted pathway? And
They can be either.
either, so it goes
Yes.
for either of the...
Yes, the exosomes are the vesicles that are in either. It's just that they're going to be releasing their contents out of the cell. Either by a regulated pathway or an unregulated pathway. Anything else? Good questions, by the way. Yes.
Look at your glasses. I'm sorry, I was wondering that since I walked in on Monday.
Really?
Yeah.
I got contacts for the first time over Thanksgiving break. I didn't want to like. Scuba diving is kind of a pain when you have glasses, and then you have to take them off, and like you don't see anything. Well, you don't see that far because it's all murky. You know, but
How bad are your eyes?
how about my eyes? I mean, they're not horrible. Like it's illegal for me to drive without anything. What's that? I don't know.
Sorry.
I don't pay attention to that really. It's like they give me the glass and I'm like, cool. But my brain, there's better things to put in there. Ethan.
When the unregulated secretory pathway dumps stuff, where does it dump it?
What's that?
Where does it dump it to?
It just dumps into the extracellular space.
Okay.
So, and usually it depends on where. Like it'll be specific. If it's a regulated secretory pathway, there's going to be specific sites that is going to dump it out of. So like, for instance, in a neuron is going to specifically dump it on the membrane, part of the membrane that is the presynaptic membrane for the synaptic cleft. There's nowhere else that has the appropriate T-snares for that to actually release into. And so that's the only place that it will dump into, yeah.
And then,
Oh, go ahead.
is it making exosomes then?
What's that?
Are they, those vesicles that it releases are exosomes?
Yes, that's going to be exosomes, correct.
For both
Yeah.
of those?
What's that? No, it's okay.
Both of those?
Both of them, yeah, for both of them, the regulated and unregulated, both can be exosomes. Okay.
How did you go scuba diving before you got contacts?
Before I got contact, I take.
Do you have a prescription mask or something?
No, I didn't have a prescription mask. I would just go, and things. I would not see anything. It'd be kind of blurry. I mean, so here's the thing: is that I take off my glasses, I could see you, and I could tell that you're human, and I could basically tell who you are, kind of. Yeah, pixelated essentially. So I would be fine, and especially. So here's the thing: is like I could tell it was you from this far away. That would be okay. Anything farther, it starts to get a little bit weird and fuzzy, and I could think that you're a small child or a girl or something like that. I wouldn't really, because it's just a fuzzy blob.
Sounds really unsafe for your diving.
But here's the thing.
Your diving visibility is
Yes.
not that far.
Yeah, see that's the thing. It's like any farther than you to me is not seeable anyways. So it wasn't horrible diving, but it still wasn't like super clear
Were you on
either.
the coast right over here? Or?
Yeah, like wait, that's north. This sounds like that way. Yes.
So where were you going diving in November?
Yeah, that's my question.
No, it wasn't diving. It wasn't in. It wasn't like I'm going diving right now. I'm hoping that I go diving over Christmas, but no, in the same place.
Where?
Like at Reserve.
If in December?
I've done it before, yeah. So the water temperature only changes by a couple degrees C. It's cold all the time. The only difference is when you get out, are you coming out into the hot sunshine, or are you coming out into snow? And then it's just a warm shower you go get into.
Have you ever considered living somewhere warmer?
I have. I've built in Florida before. I've built in Hawaii before, and that's grand and all. But here's the thing: is that invertebrates, it's colorful, but there's not much alive there. It's all clear water because they're basically deserts. And everything's kind of colorful because there's tons of light, because there's nothing growing in the water, because they're deserts. And then the other thing is there's tons of fish. There's not very many invertebrates, there's a few. I mean, there's corals and stuff, and that's cool. But octopuses, there's hardly any. Whereas here, you go diving, and there's enough invertebrate prey, and there's enough life to support a high number of predators, which octopuses are pretty voracious predators. So I've gone diving here, and I've seen 16 octopuses on a single dive before, whereas in somewhere warm, if you're lucky, you might see one.
I mean, why are you using octopuses as the standard?
Sorry, I don't understand the question.
I don't even think that needs an explanation, right? It's obvious. That's kind of like asking, why is it even worthwhile that people should continue to live on Earth? It's like, kind of nice. Do you actually need an answer to that? Okay. Okay, you guys really got me off track for long enough. Good try. So that's what happened.
Are we doing tests? Tests?
Yeah, we will. And I got a couple more minutes here. Let's talk about lysosomes, because lysosomes are kind of interesting. For 3 minutes. So, lysosome literally means hydrolytic particle in Greek. Lys means lice. Anyways, so, and they're fairly small. They're only, there's about 10-fold range in the size that they can be, from about 1/10 of a micrometer to a little over a micrometer in diameter. So they're small, but they're relatively, there's a lot of variability in the size that they have. We just talked a little bit about how they're receiving all the enzymes that they have in them, but they contain a lot. There's somewhere in the neighborhood of 40 to 50 different enzymes that occur in lysosomes. Most of these, the whole point of them being in the lysosome is that these enzymes are capable of breaking down some kind of macromolecule. Proteins, sugars, lipids, something. The point of lysosomes is to break down things. They have sometimes been referred to as the stomach of the cell, since analogies are the thing. And they're getting things from the post office, which is the Golgi, which this is becoming a very mixed metaphor here.
Yeah.
What's that? Yeah. So what's the post office of the body? So we can put it into common terms. The mouth? Like the Golgi is the mouth of the... I don't know. How about this? How about garbage disposals? Instead of a stomach, they're kind of a garbage disposal. So we're going from a post office to a giant garbage disposal. Essentially, they're breaking down things. When we talked about programmed destruction of endoplasmic reticulum, that's going to be happening via the use of lysosomes or something like that. Also, inside the lysosome, there is a very low pH. It's somewhere between 4.8 to 5.5. All of these enzymes work optimally at this fairly low pH. This also means we need to be pumping in protons pretty constantly. So we have proton ATPases that are expressed on the membrane of these lysosomes, and those are particularly important. Okay, 3 minutes. Thank you. with that, I will stop the recording.